Hi everyone, I'm Doug Whiteman from the Alzheimer's Disease Working Group, and I'm here to give you a quick update from our working group. In 2021, we published the second edition of our GWAS meta-analysis in Nature Genetics. This included around 45,000 cases, 45,000 proxy cases, and around 1 million controls. The summary statistics from these analyses, excluding the 23andMe data, can be downloaded from the CTG website. We uploaded a version with and without the proxy data. Additionally, you can apply for individual level data or other arrangements of meta-analyses through the PGC Data Access Committee. Data with 23andMe can also be accessed after approval from 23andMe. And currently, we're working on the third edition of our GWAS meta-analysis, and the primary analyst for this meta-analysis is Emil Uffelman. Our current timeline is to enact a data freeze at the end of this year, and we're especially interested in analyzing data from non-European ancestry groups. We've been able to increase the number of European ancestry cases compared to our previous analysis, and we've also added some samples from non-European ancestry groups as well. So our plan for the next year is to finalize data collection by the end of the year, optimize the proxy phenotype definition, increase the uniformity of our methods across cohorts, increase the array of sensitivity tests we do to provide more options of summary statistics, explore associations on the sex chromosomes, and perform trans-ancestry analyses. I've included a little picture and the emails of our main analyst, Emil, and as well as our working group leads, Daniela and Erla. And if you have any data that you'd like to share with them or any questions, please contact them.
This is Jack Hedema, representing the PGC Anxiety Disorder Working Group. We completed our Freeze 1 analyses earlier this year and hope to have submitted our first manuscript by the time of this meeting. Please join us at our open PGC Anxiety Working Group meeting at this conference to hear more details about these results and our future plans. The meeting will be on Thursday, October 12th from 11:30 to 12:45 in room 512G. Here are our final GWAS results. We had 122,000 cases and 730,000 controls in the final data set. We found 59 genome-wide significant loci. Our future plans, now that we've finished our analyses and our manuscript, we want to broaden our view and open up to secondary analysis proposals. We particularly invite our collaborators who have contributed all of our data that we've used and have patiently waited for these first results. In particular, we hope to collaborate with PGC-MDD, PTSD, and OCD to explore overlap in anxiety-related genetic risk. Moving forward, we want to begin assembling data for future data freezes. We want to expand beyond European ancestry cohorts. We want to focus in and analyze individual anxiety disorder data. And we want to identify samples that have dimensional anxiety phenotypes to complement those from our case control analyses. And in order to support all these, we plan to submit an NIH grant renewal for our work. Thank you very much.
Hello, I'm Ben Neal, co-chair of the PGC ADHD work group, along with Barbara Franca. And here's a slide describing what we've published in the last year, a flagship primary genetic association analysis where we've identified 27 risk loci. And you can see the Manhattan plot over here on the left-hand side of the screen. And then on the right-hand side of the screen, we actually have a series of genetic correlation analyses traversing multiple different domains of cognition. And you can see that there's a sort of overall downward shift in the genetic correlations. Most of the relationships are negative in terms of the strength of the relationship between increasing risk for ADHD and lower measured outcomes on things like spatial reasoning, verbal reasoning, attention. And that's a kind of reminder that in addition to being this clinical entity characterized by behavioral aspects, there are also cognitive performance challenges associated with individuals with these disorders, and then that some of that challenge and gap may also be rooted in shared genetics. In the last year, we've been pursuing an updated meta-analysis on the one that we published. The current interim freeze has around 36,000 individuals with ADHD and over 136,000 control individuals. That does not incorporate the 23andMe self-report information, where there are about 100,000 individuals that have self-reported ADHD diagnosis, compared to, say, 860,000 individuals in the 23andMe dataset that have not reported that particular outcome. You can see the case control breakdown over here on the right-hand side. And as with, I think, many other efforts in psychiatric genetics, we're still dominated by US and European cohorts, but we have been doing a great deal of work to try and expand and diversify those collection efforts, including cohorts from the global south, like you can see here with Brazil, Colombia represented, as well as trying to pull in some East Asian cohorts, particularly from Hong Kong and Korea. But this is still a kind of major area of emphasis and focus for us moving forwards, is to try and further improve the representation of individuals from across the world. And then finally, looking forward, most of the work in the ADHD work group is really driven by subgroups that have been spun up to take on different specific activities. You can see the sort of 5 subgroups in blue on this slide, from cognition genomics, epigenetics, copy number variation, ADHD across the lifespan, as well as the dimensional phenotypes. In particular, the dimensional analysis of ADHD has been quite useful for integrating with the clinical outcome. Our estimates of genetic correlation are extremely high between continuous measures of ADHD in the general population and the kind of diagnostic outcome that is sort of clinically diagnosed ADHD. And so this is a kind of clear area of opportunity, collaboration, and emphasis for our work group. And as with all ADHD workgroup activities, if you're interested in participating or learning more, feel free to jump onto the PGC website and connect with us. Thank you.
Hello, everyone. This is Ole Andreasen from the Bipolar Disorder Working Group. We had a good year with publications last year, focusing more on the clinical impact of genetic discoveries. And on the top left, you can see this result of the overlap analysis of cannabis use and bipolar disorder, quite extensive genetic overlap, submitted and presented here by Chang Parker. Then we have investigated the genetic landscape of bipolar disorder, both with LAVA and mixture analysis. And you can see the results of bipolar disorder here, extensive overlapping with ADHD, depression, and also schizophrenia. And also we've been working on improving clinical relevant polygenic risk scores. And here we can see the results from Choi et al. With the pathway-based polygenic risk analysis. Then the current state of the art in bipolar disorder is the current freeze. Here you can see our results from the meta-analysis with European, African American, East Asian, and Latinx cohorts. A total of 158,000 cases and 2.8 million controls. And we have close to 300 genome-wide significant loci. And here we also, in addition to the trans-ancestry analysis, we systematically evaluated the self-report data, biobank data, and clinical cohorts. And on the left hand figure, the mixer figure, you can see there is quite a lot of overlap between the clinical and the biobank and the self-reports. And finally, we have a list now of credible genes in bipolar disorder, and you can see they are associated with presynaptic and postsynaptic function. And the plans for next year is to finalize the paper You will be able to hear more details about that from Maria Coromina's presentation at the meeting. We'll have more results from the deep phenotyping task force, been very active. We have some really interesting results coming out there. And then we focus on diverse sampling, non-Europeans, and also deeply phenotyped longitudinal cohorts that's needed in a disease like bipolar disorder. So we are expanding the global participation and focused more on how to translate the genetics to clinical relevant use cases, also including collaboration with sequencing and brain imaging. So please contact us. And I would like to thank all the participants and the great team of people involved in bipolar disorder genetics in PGC. Thank you.
Hi, I'm Jordan Smoller, presenting on behalf of the Cross Disorder Group. The CDG3 analyses have examined shared and disorder-specific across 14 neuropsychiatric disorders, and we've used complementary statistical techniques to examine genetic signal at successively more granular levels of genetic architecture, from genome-wide to locus-level effects. So first, at a genome-wide level, we use genomic SEM to examine the degree to which these 14 disorders reflect higher-level latent factors of shared genetic influence. And you can see in the upper right corner, we've identified 5 latent genomic factors. The first we call compulsive disorders. The second are thought disorders or serious mental illness, neurodevelopmental disorders, internalizing disorders, and substance use disorders. And these add to the original 3 factors, those first 3 that we identified in our CDG2 analyses. We then applied Mixer to determine the degree of polygenic overlap among disorders, identifying the proportion of shared causal variants while accounting for both same and opposite direction allelic effects. Then at a regional level, we used LAVA to estimate local genetic correlations and identify hotspots of pleiotropy where multiple disorders show genetic overlap. And finally, at the level of individual loci, we used multivariate GWAS and CCGWAS to identify genetic variants with cross-disorder and disorder-specific effects. And because of limited time, I'll just summarize a few of the many interesting results. First of all, at a genome-wide level, as I mentioned, we identified these 5 correlated genomic factors. Mixer showed really substantial polygenic overlap across disorder pairs, exceeding even the overall genetic correlations, presumably due to divergent directional effects of alleles. There was convergent evidence that the internalizing factor, comprising PTSD, anxiety disorders, and depression, formed a really tight cluster of genomic sharing. At the functional and regional level, LAVA identified 458 significant regional genetic correlations that largely tracked with the overall factor structure. They included 101 regional hotspots with genetic correlations at a regional level across multiple disorders, including one region on chromosome 11 that showed regional correlation among 8 disorders. Stratified genomic SEM identified 72 functional annotations that were enriched at the level of factors. And the factors with the most significant annotations were defined by disorders with the most significant regional genetic correlations. And then at the locus level, we identified 268 loci operating through the factors, and only 35 loci that appeared to be highly disorder specific. CCGWAS identified 109 independent loci with significant differences in allelic effects across disorders. And both of these methods converged on the finding that disorder-specific variants were mainly seen between factors and not within the factors. Our CDG3 manuscript has now been drafted. We're getting input from the PGC work groups on it. And our publication is going to be delayed because we're awaiting submission and acceptance of papers from the PGC work groups that generously provided us early access to some stats. I want to give a special shout out to our key analysts who are listed there. And we have now turned to planning the next round of CDG analyses, which may include several of the possibilities listed below there, including incorporating longitudinal data to look at cross-disorder trajectories, looking at the relative value of categorical and versus dimensional phenotyping for nosology, at least from a genomic perspective, understanding and unraveling the relationship of pleiotropy to comorbidity at the phenotypic level, integrating rare variants, and further characterizing pathways that are underlying these cross-disorder factors. And that's it. Thanks very much. Looking forward to seeing everybody at the meeting.
Hi, I'm Laura Huckins, and I'm delighted to give you these updates for the PGC Eating Disorders Working Group on behalf of myself and my co-chairs Jerome Breen and Cindy Bulick. We're a large working group with investigators in more than 20 countries, and we're always delighted to welcome new members. We've made substantial progress over the last year in advancing and diversifying eating disorder genetics, and I'm showing you some of our recent publications here on the right, including new insights into the genetics of age of onset of anorexia nervosa and its relationship with age at menarche, comparisons of persistent thinness and anorexia nervosa, and discovery of heritability of avoidant restrictive food intake disorder, or ARFID. We also published a protocol paper describing our UK Eating Disorder Genetics Initiative, or EDGI. In terms of grants, we submitted a major new grant for our EDGI-2 expansion in June of this year. We received funding for 2 new large studies of ARFID in the US and Sweden, and we received new funding for our EDGI UK study, which will allow us to sequence and to create a national clinical research network. Collaborations are one of the things that make our working group so great and so productive. We are delighted to have an ongoing collaboration with the LAGC Eating Disorders Working Group and to begin a new collaboration with 23andMe for the very first time. The major focus of our working group over the last year has been an effort to expand and to improve our FREEZE-3 genetic analyses. These include nearly 40,000 cases with binge eating behavior and more than 1.2 million controls, yielding 5 genome-wide significant loci, as you can see in this Manhattan plot on the top. We've also expanded our anorexia nervosa GWAS sample dramatically. This is the largest GWAS ever worldwide of anorexia nervosa, including nearly 24,000 cases and 1.2 million controls. And this has yielded 11 genome-wide significant hits, as you can see on the bottom there. These sample sizes give us unprecedented power to investigate things like genes, subtypes, and comparisons between disorders. So I'm not showing it here, but we also have the power for the first time to look at the genetic underpinnings of, for example, restrictive and binge purge anorexia and different types of binge eating. We investigated genes associated with anorexia nervosa using a PredictScan approach, identifying novel genes and replicating some of the genes that we found in our previous anorexia PredictScan analysis. In a PredictScan analysis of binge eating, we identified an FTO gene, which will be exciting to those of you with knowledge of obesity genetics. Finally, these sample sizes also yield power to compare between disorders. So here I'm showing a case-case GWAS. Instead of just identifying genes or variants associating with case control status, here I'm showing genes that predispose to anorexia nervosa versus binge eating. So this is the first time that we've managed to actually distinguish between eating disorders. That's my 3 minutes. Thank you all very much for your time.
Hello, I'm Catherine Lewis from King's College London.
And I'm Andrew McIntosh from the University of Edinburgh. And together we chair the PGC Major Depressive Disorder Working Group of more than 500,000 MDD cases and more than 3 million controls of European ancestry. A GWAS of this data set shown on the top graph identifies 570 independent loci and polygenic risk scores, now explaining approximately 5.7% of liability to major depressive disorder. A trans ancestry update is coming very soon, but from this data, we've already identified novel cell types and drug targets that could be used for downstream studies. The polygenic prediction into unseen samples shown across the individual genotype studies in the bottom left-hand part of this slide now shows greater accuracy than is theoretically predicted by the SNP heritability. In the middle part of that graph, you see that the R² has grown with every increase in sample size in accordance with expectations. And in the far right-hand part of the bottom panel, you'll see that the prediction accuracy into clinical cohorts exceeds that into community samples. The PGC-MDD working group has also expanded into other phenotypes and into other molecular genomic measures. In the top left-hand side and the top left-hand part of this slide, you'll see that we've begun to look at GWAS studies of antidepressant response and are building on this data set right now. On the top right-hand part of this panel, you see a methylation-wide association study of antidepressant exposure, identifying new and novel loci and potentially shedding light on the mechanisms of antidepressant action. In the bottom left-hand part of this panel, you'll see a trans-ancestry GWAS of antidepressant exposure, identifying 14 loci across 4 diverse cohorts. And in the bottom right-hand part of this slide, you'll see a summary of our efforts to impute the cytochrome P450 metabolizing enzymes across PGC-MDD studies.
And here are some of our future plans. So firstly, we're going to work closely with groups with MDD studies in non-European ancestries. We have close links with PGC Africa already with Niran Okawal and Alan Kolungi. And we'll expand links with the LAGC and researchers in Asia. In addition to new studies, we'll build relevant resources and provide training. As Andrew said, our future GWAS work will be based entirely on trans-ancestry analyses, moving on from ancestry-specific work. And you can see how that's underpinned on the right, at the top figure showing how the European ancestry hits are represented in other ancestries. And at the bottom is transferability. This is far below 100% in all groups, even after correcting for sample size differences. Fourth, we'll continue our molecular epidemiology and functional genomics work. And finally, please do get involved. We're always delighted to work with people with new depression studies. Thank you.
Hi, my name is Manuel Matthijsen. I'm one of the co-chairs of the TS-OCD working group, and I'm here to present the updates for the OCD section of the group. With regards to publication, 2022 and early in 2023 were slow years for OCD. Here to report one paper from a secondary analysis proposal, which is listed first, an update to a submission to medRxiv about our current work, and other than that, 2 publications from work that has been done in the subgroup for hoarding and OC subtypes. With regards to the current state, we have finally submitted our manuscript for the latest freeze, which includes 53,000 cases and about 2 million controls, including data from 23andMe. We report 30 genome-wide significant loci, including previously published GWAS hits for OCD. In total, some 25 genes are indicated by functional and positional analysis, and we analyzed 4 broader subgroups, clinical, biobank, comorbid, and self-report data to look further into heterogeneity of our signals. We find that a single factor explains the majority of variability seen in the GWAS, and that the lowest factor loading is at around 80%, stemming from biobanks. The strongest genetic correlations overall are with phenotypes and traits out of the anxiety, neuroticism, and depression spectrum as expected. But we also see the strongest heterogeneity of correlations among the 4 broader subgroups mentioned before for the very same groups of phenotypes and traits. Since we are now looking into using the latest freeze, we welcome new secondary analysis proposals, and we're already seeing a huge increase in numbers for those. Existing proposals currently focus mostly on cross-disorder topics, that is OCD with 1 or more other phenotypes using, amongst others, genomic SAM models. Our next OCD GWAS freeze is certainly looking into diversification of contributing samples with contributions from the Latino study, MVP, GPC, and others. We also would like to include a larger sample from third parties, which currently account for around 150,000 cases, but it's yet to be seen whether we can be successful in that. And of course, we would like to refine our current interpretation with regards to heterogeneity. With regards to our subgroups, VOC symptom subgroups is increasing their sample size and now also including case cohorts. The treatment response working group has now started to do their first analysis. And in terms of our CMV work, the first analysis freeze is also around the corner. Thank you very much.
Hi, Aidan Corvin is my name. I am the PI for the Pedigree sequencing project in PGC. Just to remind people what the purpose was, it was really to identify and analyze large, densely affected pedigrees with multiple psychiatric disorders and not exclusively necessarily the same disorder. And we wanted to contribute as part of that, obviously, to developments in methodology and pipelines for this type of analysis work. And longer term goal really to collate the data and get a better overall picture of the role of pedigrees and rare variants in pedigrees within the structure of these disorders. So I think we have developed streamlined processes for data processing and analysis and published on that, and we've applied the methods to look at CNVs, single nucleotide variants and polygene scores in pedigrees, and we've learned a lesson really that different pedigrees require different approaches. So just to review here, progress to date, as you can see, there's almost 500 or 450 or so sequences that we've been looking at across various colleagues. We've really published on where we found evidence for rare deleterious mutations, and probably best evidence so far was the large Tourette syndrome pedigree that we worked on with Carol and colleagues, published in Molecular Psychiatry. Gives a good sense of really the type of work we've been trying to do. And we currently have work on a schizophrenia pedigree with Bill Brierley that's under review, and the MZ twin pairs that we've been working on with Pat. And we have there's some other pedigrees that we're interested in looking on that aren't on that, aren't included there. So apologies for that. I think what we've been, one of the interesting things is working with Mohammed and his colleagues is a pedigree where there's strong IBD evidence for haplotype sharing, but no clear candidate mutations. And we've been trying to work through that a lot recently, and we'd hope to draft, we're in the process of drafting a paper around that. And then clearly we've seen pedigrees where there's no evidence of excess IBD sharing, but increased evidence of PRS, suggesting sort of common variant profile there. And in our work to date, we've really found no evidence for involvement of the known risk CNVs in any of the pedigrees that we've looked at. So with respect to our planning and where we want to go from here, the team is small. Many of you know Michael, who has been in the community a long time and is now retired. I think we've done a lot of work to refine our processes and what we're doing. We're happy with the structures and the pipelines that we have. We'd like to complete our existing pedigree data and archive it. But certainly, I think at this stage, we'd really like to expand the group, invite other people to join us, maybe have a better profile on the PGC website, and really just canvass other people who are trying to do similar work in the community and have sort of a stronger, wider reaching working group to collaborate more. I think this will give us a better picture of what's actually happening across the pedigree structures, across disorders, and it'd be nice to integrate the data with colleagues who have this type of data. So I think our approach really from now on is looking outwards more, and I think that, as I said, we really welcome collaboration. Just for the WCPG meeting, for personal reasons, I'm not going to be there this year, but my colleagues Niamh and Cathal will be there and be delighted to talk to anyone, and that's my email, so please reach out. So thank you and enjoy the meeting.
Hello everyone. It is my pleasure to present on behalf of the PTSD working group. The last year has been very successful for us. We have two primary publications to report. First, in collaboration with the CNV group, we have published the first rare copy number variation in PTSD. And second, the PTSD Freeze-3 paper, in which we describe 95 PTSD loci, is currently under review at Nature Genetics. In addition, we have supported several secondary publications with our summary data and our collaborations with the 11 working groups. Our current results have been posted in medRxiv. They are based on a multi-ancestry GWAS including over 1 million participants with over 150,000 PTSD cases. We describe 95 unique risk loci and a comprehensive set of post-GWAS analysis to prioritize PTSD genes. If you want to hear more about it, join us in the PTSD Symposium this Friday at 3 p.m. Our plans for the year ahead include, most importantly, the R01 renewal for our working group. We are also starting the collection of data for Freeze 4, including the integration of common and rare variants. We have a focus on admixed populations, including the implementation of Tractor, our optimized local ancestry pipeline, and a focus on polygenic risk scores. We will provide leave-on-out summary results for all of our cohorts and try to optimize PRS for admixed individuals. Most excitedly, together with our East African collaborators, we will start a newly funded R01 initiated by Kerstin Köhnen to investigate genetics of PTSD in African populations. Thank you.
Hi everyone, James Walters here on behalf of the Schizophrenia Working Group of the PGC. It's my pleasure to speak for the group. Firstly, our outputs. It's been really pleasing to see that the Nature paper from last year has continued to garner a lot of interest. I think one of the most prominent and important papers out this year was the output related to the PGC3 targeted sequencing study led by Alex Charney and colleagues at Mount Sinai. So congratulations to them. And then we've continued to produce work related to the biology, the functional genomics, but also methodological development. And I think that's key. And we're really happy when the data has been accessed for that reason. So thanks and congratulations to those researchers. What we know, I should thank my co-chair, Mick O'Donovan, for putting these together, and I'll leave you to read those. I want to concentrate on the bottom part of that slide, which, as well as what we're starting to learn about mechanism and the tissue and cell types that are involved, as well as getting further phenotypic insights, then we're beginning to think in detail about how we expand our samples further. The results of one such effort, led by Cynthia Balangero, who's just done a great job since she's been in place as the Latin American representative of the group and conducted this survey. And this has shown us that there continues to be a lot of interest among non-genomic researchers who are the respondents to this survey, and also that there's untapped resource, but actually there are important barriers, and that is access to genotyping. So there are, there's DNA out there to be accessed in that respect, but also resources for further recruitment and a need to support training analytically. It's great to see the work that's been done at this conference in that regard, but we need to continue to do it. And then the other thing that came out consistently was about a lack of ideally local, but certainly independent funding to support this work. And hopefully that's informative as we start to think about these next stage developments, and others are working in this sense already. And finally, plans for the coming year. Well, there are several efforts that we want to push over the line. We'll hear about the HLA analysis at the conference. We also need to think about taking forward the schizophrenia bipolar analysis. We want to be able to bring together the large samples that are out there. And I think our continental representatives, we now have appointed people across the world to be group representatives, and I think they've really hit the ground running and have been a real asset for the group. So thanks for their efforts. And we also want to be able to access some of the other large samples that are coming online, but perhaps haven't been traditionally diagnosed and are using new methods of diagnosis. And so we've brought together a group to think about those methods and make recommendations around that, because we also want to incorporate them in a judicious way. And then finally, there are new groups being set up all the time. A first episode psychosis group with longitudinal data that I think will prove of real value. And then lastly, a treatment-resistant schizophrenia group. We've got a new grant in that, and that's a field that could prove important for precision psychiatry going forward. Finally, just to make a plea, whoever would like to hear more We've got a series of flash talks as well as update information at our group meeting on Thursday, the 12th of October at 11:30 in room 510. Cheers and goodbye.
Hi, everyone. The Substance Use Disorders Working Group of the PGC is funded by the National Institute on Drug Abuse. Last year, we published 2 papers. Of these, the Hatum et al. Study in Nature Mental Health was accompanied by a joint press statement from NIDA, NIAAA, and NIMH, and is this year's Gershon Award recipient. Congrats, Alexander. Our data were also part of 2 accepted Million Veteran Program publications on alcohol and cannabis. In addition, our early career investigators that are part of our team have also received their own funding via career development awards and independent investigator grants, and we thank them for their really outstanding contributions to the PGC SUD group. As before, our work continues to show genetic differences between substance use, so something like how often or how much somebody drinks on a typical drinking day, and substance use disorders. The Hatum et al. GWAS also showed that, very consistent with prior twin studies, a large number of genetic variants exert their influence on liability to problems with multiple substances, and that the substance-specific variants are primarily receptor-specific or related to drug metabolism. We also found that adjustments for substance use genetics only modestly impacts these results and did not change associations, for instance, with genetic correlations with psychopathology. Extending our study of heterogeneity, we have now undertaken a GWAS of DSM nicotine dependence and are finding it to be far more genetically correlated with other substance use disorders as well as psychopathology, as compared to other indices of heavy smoking or GWASs of the Fagerstrom test of nicotine dependence or cigarettes smoked per day. We also continue to document the need for more well-characterized SUD data. Our plan for the next year is obviously to submit our DSM nicotine dependence GWAS, which we're kind of wrapping up at this point. We are working to bring in data from the All of Us study to add numbers to our existing substance use disorders phenotypes, and we want to again invite members of other PGC working groups to share any comorbid substance use disorder cases and controls with us if possible. Finally, supported by our publication that no amount of omics data can substitute for increased sample size, we used carryover funds from PGC3 to genotype additional SUD cases, which are almost ready for the next round of analyses, so stay tuned for our report on that. Thanks so much for listening, and we invite you all to join us at our open meeting on Thursday from 11:30 to 12:45. Enjoy WCPG. Thank you.
Hi, this is Anna Doherty speaking on behalf of the PGC Suicide Working Group. Over the last year, we've released a multi-ancestry GWAS meta-analysis of suicide attempt in about 44,000 cases across 22 international cohorts. This included meta-analyses of specific ancestry admixtures, and this is currently in press at the American Journal of Psychiatry. Other publications include a multi-ancestry GWAS meta-analysis of suicide attempt and also of suicidal ideation in the Million Veteran Program. These both stemming from, all of these works rather, stemming from hard work by Nathan Kimbrell, Allison Ashley Koch, Jeannie Beckham, and others on MVP. Summary statistics for all of these analyses can be obtained through PGC, through the PGC Data Access Portal or through dbGaP for the Million Veteran Program. So there are some new insights from these studies. For one, we observed strong signal in an intergenic chromosome 7 locus, stronger for suicide attempt than for psychiatric disorders, which is also, this locus is also associated with phenotypes like risk-taking, smoking, and insomnia. We observed strong genetic overlap of suicide attempt across civilian and veteran populations. We also observed significant genetic overlap across suicidal ideation, attempt, and suicide death. And the last thing to note here is significant enrichment of suicide attempt risk in genes targeted by psychiatric medications and genes expressed in brain tissues. So our aims from last year were to increase the diversity of genetic data on suicide, and we're going to continue working very hard on this. We now have a total of 40 cohorts committed, and we are accepting both individual level data and summary stats from these cohorts. To streamline the inclusion of cohorts, we're providing analytic support as well on a case-by-case basis, both for GWAS and for any site-specific analysis goals that are in line with the working group's goals. Please do contact us if you're interested in collaborating. We're implementing a new phenotyping protocol as well, including formal case control definitions using multiple measures and methods. And this includes lists of ICD codes that we have validated in Vanderbilt and VA cohorts. So we're also continuing data collection in India with the goal of creating a blood and brain tissue biobank for the study of population-based suicide death. We're really excited about this partnership with PI Chita Behera and his team in Delhi. So for extended updates and to learn more, please join our in-person group meeting this Wednesday at 11:30. And directly after that, a talk by Sarah Colbert reviewing the working group updates in more detail. And we have a call each month, and you can reach any of us chairs by email if you'd like to join in. That's myself, Neve Mullins, and Doug Ruderfer. Thanks so much.
The PGC TS Working Group has had another productive year. The 7 papers listed here were all led by trainees since the last World Congress. Of note, these trainees represent 6 different PGC TS sites. In terms of scientific progress, we have updated our final freeze of the TS GWAS-3 analysis with 13,247 cases and 536,000 controls. As you can see, we now have 6 independent genome-wide significant loci, which we are currently replicating in 1,500 TS cases and 941,000 controls from 23andMe. In addition, Sheng Wang and Matt Halverson have completed the TS-CNV3 analyses in 5,726 cases and nearly 11,000 controls. They demonstrate a significant increased burden of CNVs spanning known neurodevelopmental disorder genes as defined by Fu et al., Nature Genetics 2022, as well as neurodevelopmental CNVs found in the UK Biobank identified by Kendall et al. That was published in Biological Psychiatry in 2017. In terms of individual CNV loci, proximal deletions of Nerexin-1 were again identified as genome-wide significant, as well as duplications of ACACA and the larger 17q12.1 duplication. Regarding plans for the coming year, we expect to complete sex-specific GWAS analyses, epigenomic and transcriptomic enrichment studies, as well as pathway and network analyses. Perry Peshu is also leading an Enigma TS Imaging Genetics Consortium effort. In addition, in terms of global diversity aims, there are ongoing collections in Korea, led by TIC Genetics, and small cohorts of TS patients from Latin America, though additional large-scale collections are needed. As well, we welcome any questions, as well as additional collaborations. Thank you.